It's very faint. If you could try to move closer, and please proceed with the first slide, Frank.
Yep, one second.
Lucio, if you could try and move closer to the in the speaker, that would be great. Thank you so much.
Difficult connection. Can you hear me?
Yes, please proceed.
Okay. I'm sorry that I did not see and hear much, but I heard the last talk from Christopher Happy, and I wish to congratulate him for the very important work done in Nigeria, where I lived many years ago. And if I can have the next slide, please. You all know what coronavirus looks like, so I will go very quickly through the first few slides. This is a micrograph of the virus, and the next slide, please. The next slide shows a structure of the virus. The spike. Now, the membrane, which is largely borrowed from the host, and the RNA genome and protein inside. Next slide, please. This is a very important step when the virus enters the cell. It is, of course, absolutely crucial. We know that the ACE2 is the receptor for the virus, so there must be a very specific recognition between the receptor and the spike of the virus. When the virus gets to the cell, it gets to the cell by endocytosis. These steps are very important, obviously, for the biology of the virus, but they're also highly relevant to what could be future therapeutic approaches. It is clear that every one of these steps is necessary for the virus eventually to fuse with the lysosome and to then start the replication process. The characteristics of the RNA viruses are very, very distinct. Here I excluded 5 of them, all of which are familiar to you and very important in human pathology. As you know, RNA viruses are of 3 types. Some of them, like the HIV, are retroviruses. The first thing that happens upon infection is that a cDNA copy of the virus is made, and then starts the replication process. Others, like the Ebola, the so-called minus strand, that is the RNA which is non-coding, and the plus strand is the first thing that is made, and then modified then for the viral process. COVID-19 instead is a virus with a plus strand, meaning that the RNA in the virus is directly the mRNA that is translated into protein. Each one of these viruses has the characteristic cell that is able to infect because they have the appropriate receptor. So remember, receptor, the endocytotic process, and then the RNA that is expressed by hijacking the ribosome of the host. The virus is able to displace all the RNA of the cell from the ribosome and forces the ribosome to make its own protein in form of a polyprotein. And the polyprotein must then be protease split by specific proteases to give the proteins that constitute the virus. Each one of these steps is potentially a step that could be druggable, as we variety of changes that have occurred since the first, what could be regarded as case zero, which, as you know, we think is the species jump from bat to human. But since that time, presumably before December of last year, there's been literally a micro or micro evolution of the virus, which has produced a variety of different sequences. As you see in this slide, they're color-coded, and they're color-coded mean where these various sequences have been found. Now, there is no question that the original species jump must have taken place in China. So you can see a large disk in China and then different colors. What does it mean? That already in China, there were different forms of the virus, which for simplicity we can call clades. And as you can see, members of the clade in blue-green apparently migrated to Europe.
Frank, I think we're on the next slide. Dr. Major, please go to the next slide.
Orange red are those that migrate to the United States across the Pacific. So very interesting that there are different species, literally different sequences, not species, but different forms of the virus in different parts of the world, and probably it continues to evolve. Next slide, please. So going from the biology to the epidemiology, these are the entities that we need to consider, and that, as you all know, now are widely reported in the literature and also in the daily newspapers. There is a difference between persons infected but asymptomatic, infected with symptoms, infected with severe disease, and unfortunately those who have died. And also by now, we have thousands of people who have recovered. And I think this is very important when we think of therapy.
Sorry, Frank, I think you need to move to slide 12. This is the figures that are important in an epidemic. Yes, I'm terribly sorry about this, participants. Yeah, we're on this slide. Lucio, we're on the slide that's talking about the numbers of infected persons infected, etc. Just for people to understand from an epidemiological point of view the meanings of the different numbers. Thank you.
Sorry, I couldn't hear this. Should I continue?
Yes, please continue. We were catching up with the slides with the number, the figures that are important in an epidemic, the number of persons infected, etc., etc. That's the slide that is being projected. Okay.
So I wanted to, as you know very well, Italy has been very hard hit by the virus. I think I'm not an epidemiologist, and I'm not a virologist either. I'm only a hematologist. I'm, of course, keenly concerned with what has happened in Italy, and I really hope that it should not happen elsewhere. Now, we don't know why Italy was so hard hit, but I think it's important to emphasize the great contagiousness of this virus. Nobody knows as yet how many viral particles you need to cause infection, but I suspect that the high mean that very few viral particles are sufficient to cause the disease. In Italy, on the 19th of February, there was a major football match between Atalanta and Valencia of Spain. There were 1,000 spectators. And the match took place, the Atalanta match is the team of Bergamo, but the match took place in Milan. Within 3 weeks, Milano and Bergamo were the cities largest number, as you know, where the epidemic has had its epicenter. And so later, about the same time, in Spain, as you know, there was also a very high number of cases. So I think contagiousness is enormous, and you must consider these chance events where large crowds come together that could be crucial. I call them stochastic events. And here you can see the curve of very up to date, I think it's up to the 6th of April. And we hope Italy is, people say, past the peak or on the cutoff. If you look at this curve, unfortunately, we cannot say that, but we hope that this is the case. Next slide, please. There's been a lot of discussion about the effect of temperature. We hope that where it's hotter, the virus.
Next slide, Frank.
That may be true to some extent, but the effect is very small.
Next slide.
Next slide, please. Another important point is an interesting observation that countries where there has been a policy of BCG vaccination have had less cases than countries where there has been not this policy. It is possible that effect of vaccination, like BCG, in a way activate the immune system in a way that is beneficial. In this respect, very, very interesting is that recently I read a report, very recent report of a patient who had renal transplant and depressed immunity and yet a very mild COVID-19 infection, indicating that the immune reaction very particular, and it's not good enough to say you have high immunity or low immunity. It is something much more subtle. Next slide, please. We can skip this one, and I want to conclude by a summary of epidemiology versus management. Clearly, there are different tasks. One is prevent infection. This obviously is the primary purpose of all the restrictive measures that different countries have taken. Reduce the time during which infection to others. It is not yet clear how people can do that. Avoid in infected people the transition from mild form to severe form, heralded by ARDS, acute respiratory distress syndrome. And as you know, there are hopes and current trials as to anti-inflammatory drugs may be beneficial in that respect. This would be very, very important. Another item that has not yet been just begun to be explored is very important. It is the potential use of plasma from convalescent or rather recovered people, people who are cured of COVID-19. As I told you, in Italy now there are probably about 20,000, and these people must have antibodies. And experiments are important. This is a challenge for blood banks to collect their plasma that could be used for actual passive immunization, that is, to protect from serious disease people who have contracted it. And of course, the treatment of ARDS has to do with intensive care to save the patient's life. And of course, we should protect the contact and specifically health professionals. So I don't want to take any more time except to say that I hope to come back to Tanzania very soon. And it is very important that I hope very much that the contact tracing will be successful. I hope very much that the disease will be confined and that it will not reach carrier cost. very much. Again, I'm not a virologist on epidemiology, so forgive me my trespasses and give you a hematologist view of this very difficult problem.
Thank you. Thank you very much, Professor Lucio. So I do thank Professor Lucio for his presentation. This is tremendous, and we'll have to share the experience of Zoom meetings for participants who have no internet access, such as Lucio in Genova. And then we'll share the slides that are available, and we'll try and address the questions that have come up from all the speakers. Because of time, I'd like to invite Dr. Bira and Mariam to talk about COVID-19 and sickle cell disease. And